You know, if you happen to live an average human lifespan, you are going to spend roughly 25 years of it completely, totally, and utterly unconscious.
It is a staggering number when you really think about it.
Right. Like, I really want you to just sit with that number for a second. 2.5 decades of your existence just gone to the void.
Just entirely wiped from your subjective experience.
Yeah. You are lying there, paralyzed in a dark room, hallucinating wildly and completely vulnerable to the physical world around you.
Which is terrifying from a biological perspective.
Exactly. I mean, from a purely evolutionary standpoint, doing this every single day sounds like an absolute disaster. It sounds like, the easiest possible way to get eaten by a predator.
Without a doubt, it is a massive evolutionary risk.
And yet this daily excursion into the dark is so non-negotiable, it's so deeply hardwired into our biology that your brain will literally force you into it if you try to resist. Like, it will shut you down against your conscious will.
You cannot fight it indefinitely. The brain always wins that battle.
It always wins.
Mm-hmm.
But here's the part that I find deeply unsettling. Despite this state consuming a full third of our existence on this planet, the exact fundamental biological functions of sleep still remain a subject of intense scientific debate.
Oh, it is arguably one of the greatest enduring mysteries in all of modern biology. I mean, we know with absolute certainty what happens when you don't do it.
Right, everything falls apart.
Everything. Your cognition fails almost immediately, your memory degrades, your metabolic systems go entirely haywire, and eventually, total sleep deprivation is fatal. But the fundamental why we do it and the mechanical how of the process are incredibly complex.
And people have been arguing about this for centuries, right?
Oh, absolutely. Historically, the scientific and philosophical communities have swum wildly between different extremes when trying to understand this. If you look back at Plato or Aristotle, or if you jump forward to the late 19th and early 20th centuries with someone like Sigmund Freud, the prevailing theory was always that the dreaming component of this state carried deep, profound personal meaning. It was viewed as the subconscious trying to communicate, like a psychological treasure trove.
Right. That is the whole dreams are the royal road to the unconscious idea. That belief that every flying dream you have or every dream where your teeth fall out meant something highly specific about your childhood trauma or, your repressed desires.
Exactly.
It was all about finding the hidden psychological signal in the noise.
And that was the dominant paradigm for a really long time. But then you look at modern pioneers of molecular biology, people like Francis Crick, who was the co-discoverer of the structure of DNA.
A heavy hitter.
Very much so. Crick looked at the phenomenon of dreaming and hypothesized the exact opposite of Freud. Crick speculated that dreaming is quite literally the brain throwing out its trash.
Wow, just taking out the garbage.
Precisely. Under his framework, it is the neurological equivalent of emptying the recycling bin on your computer. It's a mechanism for clearing out the parasitic bits and pieces of daily experience, the random synaptic connections that simply aren't worth retaining.
The network doesn't just overload.
Right, so it doesn't become overloaded. And today, while many researchers focus heavily on the idea that a core function of sleep involves synaptic remodeling and the active consolidation of the memory traces you form during the day, the specific evolutionary role of the dreams themselves in that cellular process is still hotly debated.
So we have this massive 25-year-long mystery, which brings us to our mission for today. We are embarking on a special 2-part deep dive. And I want to set expectations right up front for you listening. Consider this your advanced masterclass.
We are getting very technical today.
Yeah, we are not just going to give you standard sleep hygiene advice about putting your phone away or like how to get your 8 hours. We are going straight into the dense, serious neurological architecture.
The actual wiring.
The wiring. We are going to unpack the physical wiring of how your brain actually puts itself to sleep, how it wakes itself back up, and the incredibly intricate biochemical and genetic clocks that regulate that entire process. And we are pulling all this rigorous detail directly from the foundational source, which is Chapter 44 of Principles of Neuroscience.
And to really understand this landscape, we cannot just rely on subjective reporting. You can't just ask people how they slept.
Because people are terrible at knowing how they slept.
They really are. So we have to start by looking at the objective tools used to map it. When researchers or clinicians study this state, they rely on a polysomnogram.
Okay, the polysomnogram.
Yes. This is the absolute gold standard for sleep research, and it relies on 3 core physiological measures to give us a real-time picture of the nervous system.
Break those down for us.
First, you have the electroencephalogram, or EEG. This records the electrical field potentials generated by the cerebral cortex. This is what we colloquially call your brain waves.
The classic squiggly lines on the monitor.
Exactly. Second is the electroencephalograph, or EOG. This records your eye movements using small electrodes placed on either side of the face near the eyes.
Which, I have to say, feels a bit counterintuitive at first. Like, you are asleep, your eyes are closed. Why do we care what the eyes are doing?
It seems odd, yes.
But what your eyes are physically doing behind those closed lids turns out to be an incredibly important diagnostic marker for what stage of sleep you are in, right?
It is absolutely essential. You cannot accurately stage sleep without it. And then the third measure is the electromyogram, or EMG, which records muscle fiber firing.
So that's checking muscle tension.
Right. We usually measure this through the skin under the chin to determine your overall skeletal muscle tone. Now, if you were doing a clinical polysomnogram in a hospital setting to diagnose a problem, you would also continuously measure respiration.
Like tracking breathing.
Yes. Airflow, chest wall movement, blood oxygen levels. Because breathing is frequently and sometimes dangerously disrupted in many clinical sleep disorders.
Which we are going to explore deeply in part 2 of this deep dive.
We will. But for understanding the basic architecture, those three electrical measures, the EEG for the brain, EOG for the eyes, and EMG for the muscles, those are the essential trinity that allows us to see the invisible internal structure of the state.
I really want to dig into that structure now. Because it is not just one uniform block of darkness, is it?
Not at all.
It's this highly dynamic, alternating architecture of non-REM and REM sleep. So let's map the territory. If we are watching those three monitors, the brain, the eyes, the muscles, what actually happens electrically as we transition from just sitting here wide awake and alert down into the absolute deepest levels of unconsciousness?
Well, let's start with the baseline default state, which is active wakefulness. If we look at your EEG right now, it is characterized by very high-frequency, low-voltage electrical activity.
But it's very fast and very small.
Yes. If you look at the readout on a screen, it essentially looks like static.
Just a messy blur of line.
A very messy blur. And the physiological reason it looks like static is because billions of individual cortical neurons are all firing away at different times, doing their own unique, independent processing. At the same time, your EOG shows your eyes darting around the room taking in visual information, and your EMG shows variable, moderate muscle tone because you're holding your posture, making small movements, resisting gravity.
So the brain monitor looks like static because it is like a crowded room at a party. Everyone is having their own individual conversation. There is no unified chant, just a loud, busy, high-frequency hum of localized activity.
That is a very accurate way to visualize it. It is desynchronized activity. Now imagine you get into bed, you close your eyes, and you relax into a state of quiet wakefulness.
Okay, getting comfortable.
Almost immediately, the electrical pattern changes. Rhythmic waves start to appear, particularly over the occipital region at the back of the head.
Because that's the part of the brain that processes vision. Right?
Exactly. Since you've closed your eyes and cut off visual input, that visual area starts to synchronize. These are called alpha waves, and they oscillate at about 8 to 13 hertz or cycles per second. You are still fully awake, but the system is beginning to synchronize its firing.
And then we drop in.
Yes. From there, you cross the threshold into stage N1, which is the lightest stage of non-REM sleep.
The entry point. Like the shallow end of the pool.
Yes, the very shallow end. In stage N1, the EEG slows down even further into what we call the theta range, which is 4 to 7 hertz.
Slower waves.
Slower waves. Your eye movements also slow down to these very gentle, slow rolling or roving motions under the lids, and your muscle tone drops slightly.
Are you actually asleep here?
Consciousness is definitely beginning to fade, but you are still very close to the surface of waking reality. If someone spoke your name quietly or a floorboard creaked in the hallway, you would be awakened easily.
Yeah.
And frequently, people woken from stage N1 will swear they weren't actually asleep yet.
Oh, I know that feeling perfectly. That is the classic dad in the recliner defense. I wasn't sleeping, I was just resting my eyes.
We hear it all the time.
But scientifically, the objective brainwaves on the EEG tell a completely different story. They were definitely asleep. So we slip past N1, deeper into the pool. What happens when we hit stage N2?
As you descend into stage N2, the background electrical activity continues its general slowing trend. It operates mostly in the theta and delta ranges, so we are talking between zero point 0.5 and 4 hertz now.
Getting very slow.
But what really defines stage N2, what a sleep technician is actively looking for on the screen, are two highly specific, explosive electrical events that punctuate that slow background.
Okay, what is the first one?
The first phenomenon is called a sleep spindle.
A sleep spindle?
Yes. These are sudden, intense bursts of 10 to 16 hertz activity. They wax and wane over the course of about 1 to 2 seconds.
So they just pop up out of nowhere?
They do. And if you look at them on the physical EEG readout, the squiggly lines literally resemble an old-fashioned yarn spindle.
Like tapered at the ends?
Exactly. Tapered and narrow at both ends and thick and wide in the middle.
Okay, and what is the second signature event of stage N2?
The K-complex.
K-complex.
This is a massive, high-voltage, single slow wave that just suddenly erupts on the EEG tracing, often followed immediately by one of those sleep spindles.
It's a giant spike on the graph.
A massive spike. A K-complex is actually one of the largest electrical events that the healthy human brain produces.
Wow.
During stage N2, those slow rolling eye movements from N1 usually stop completely, and you are generally unconscious of the world around you at this point.
And from there, we drop all the way down into the basement. Stage N3, the deepest end of the pool.
Stage N3 is the deepest state of non-REM sleep. It is completely dominated by abundant, very slow, high-voltage delta activity.
Delta waves.
Yes. Your brain waves are sweeping back and forth at less than 4 hertz, often between 0.5 and 2 hertz.
That is incredibly slow.
It is. Across all these non-REM stages, but especially here in N3, your eye movements have stopped entirely. Your muscle tone remains very low, your breathing becomes incredibly slow and highly regular, and your core body temperature falls.
So the body's just fully shutting down operations?
Effectively, yes. In stage N3, you are profoundly, deeply unconscious. That massive, slow cortical electrical activity effectively blocks external sensory information from being processed by the higher brain.
So you wouldn't hear the floorboard creak anymore?
No. It is incredibly difficult to wake someone from stage N3. And if you are forced to do so, like by a loud alarm, they will exhibit severe sleep inertia.
Like that awful groggy feeling.
Exactly. They will be extremely groggy, confused, and disoriented for several minutes because their brain was so deep in that delta rhythm.
Okay, I have a hard time wrapping my head around what those squiggly lines actually represent in physical space. I mean, we see these slow, massive delta waves sweeping across an EEG monitor, but an EEG is just reading electrical fields through the skull and the scalp.
Correct. It's measuring field potentials.
Right. So what is physically happening to the actual individual neurons inside the cerebral cortex to create these giant rhythmic waves? Like, how does the busy, static-like chatter of the awake brain, that credit party we talked about, turn into this massive synchronized ocean swell?
It's a great question. To understand that, we have to look at the deeply technical cellular mechanics of cortico-cortical and cortico-thalamic interactions.
Okay, let's get into the mechanics.
Let's focus on the pyramidal neurons in your cerebral cortex. These are the primary excitatory neurons of the brain. During these deep non-REM stages, the membrane potentials of these millions of individual neurons stop acting independently.
They stop the individual chatter.
Right. Instead, they start fluctuating in a highly synchronized, rhythmic way between what we call up states and down states.
Up and down states. What does that mean for a single cell?
Well, an up state is a period when the neuron is depolarized.
Meaning it's active.
Yes, it is electrically active, receiving inputs and firing off action potentials. A down state is when the neuron becomes heavily hyperpolarized.
So it goes negative.
Yes, it is pushed far away from its firing threshold, rendering it completely silent and temporarily incapable of firing at all.
So they alternate.
Exactly. So imagine millions upon millions of cortical neurons are firing together in an up state, and then a fraction of a second later, they are all falling totally silent together in a down state over and over again. That massive, unified, synchronous firing and the subsequent unified silencing produces huge, oscillating waves of electrical potential across the dendrites of the cortex. And that immense, synchronized shifting of voltage is exactly what the EEG picks up on the scalp as a slow delta wave.
Okay, so if the awake brain is a crowded room where everyone is having their own individual conversation, deep sleep is like a massive stadium full of people doing the wave.
Ah, I like that.
Right, like everyone stands up and yells at the exact same time. That's the up state, and then everyone sits down in total absolute silence at the exact same time, which is the down state. and they just keep doing that rhythmically across the whole stadium.
That is a very helpful way to conceptualize the synchronization, and the mechanism behind those strange bursting sleep spindles we see in stage N2 is even more intricate than that.
How so?
Well, it involves a very specific chemical conversation between the cortex and the thalamus. The thalamus, as you may know, is the brain's primary sensory relay station.
Right. Almost all sensory information from the body passes through the thalamus before it reaches the cortex.
Correct. Spindle generation specifically involves neurons in a thin sheet called the reticular nucleus of the thalamus and the thalamocortical relay neurons themselves.
Let me make sure I'm tracking the anatomy here. The thalamocortical relay neurons are the ones that actually pass the sensory data like touch, sound, etc. up to the cortex.
Yeah.
And the reticular nucleus is a nearby structure that acts like a manager governing those relay neurons.
Yes, that's exactly right. Now, during non-REM sleep, the thalamocortical relay neurons are generally hyperpolarized.
Held in that down state.
Right. They are inactive, which makes logical sense because you are asleep and you shouldn't be processing external sensory input.
Makes sense.
But the neurons in the reticular nucleus will periodically send inhibitory signals down to those relay neurons. This drives them even further into hyperpolarization.
Pushing their electrical charge even deeper into the negative.
Correct. And this extreme hyperpolarization actually triggers something wonderfully counterintuitive in the cell membrane. It causes a specific type of ion channel, low-threshold calcium channels, to suddenly open.
Wait, going deeply negative opens the channel.
Yes. Because of the concentration gradients, calcium ions then flood into the thalamocortical neuron. This sudden influx of positive calcium ions drives a rapid, intense burst of sodium spikes.
So the cell just fires wildly.
Exactly. The relay neuron suddenly fires a rapid burst of signals. Those signals travel back up and excite the reticular neurons, causing them to fire again, which initiates the exact same cycle all over again.
It creates a microscopic chemical feedback loop.
An excitatory-inhibitory feedback loop, yes. This pattern of firing and inhibiting repeats roughly every 100 milliseconds, which is what produces that 10 to 16 hertz frequency we record on the EEG as a spindle.
Oh, wow.
But the loop can't sustain itself indefinitely. After a second or two, the reticular neurons become less responsive to the feedback, the loop loses its momentum, and the spindle electrical activity wanes.
Tapered at the beginning as it ramps up, thick in the middle, and tapered at the end as it dies out. Just like you see on the monitor.
Just like the yarn spindle.
That is incredible. We're talking about a microscopic chemical feedback loop of calcium and sodium ions deep inside the brain, generating a specific electrical wave that washes over your entire cortex.
It is a beautiful piece of biological engineering.
Okay, so we've mapped the descent into deep, quiet sleep, the stadium wave. But about 90 minutes into this cycle, something radical happens.
Yes, a complete shift.
In 1953, two researchers at the University of Chicago, Eugene Azarinsky and Nathaniel Kleitman, noticed that their sleeping subjects would suddenly start exhibiting these jerky, rapid conjugate eye movements under their eyelids.
The EOG monitors were going crazy.
Right. They woke the subjects up during these specific periods, and roughly 75% of them reported vivid, immersive, highly visual dreams. They had discovered REM sleep, rapid eye movement sleep. And this stage completely shatters the peaceful architecture we just spent all this time describing. So how does the brain get out of that deep stadium wave rhythm to start dreaming?
The transition into REM sleep is a profound physiological shift. When a person enters REM, their EEG suddenly changes. It abandons those slow, massive, synchronized delta waves and reverts back to fast, low-voltage, desynchronized activity.
It goes back to static.
Yes. If you just looked at the EEG, the brain looks almost exactly like it does when you are wide awake and highly alert. Your cortex is incredibly active, processing internally generated visual and emotional information.
But physically?
Physically, the rest of your body undergoes an extreme, almost dangerous transformation.
Right, because if your cortex is that active, if you are running from a monster or flying through the air in a dream, why aren't you getting out of bed and physically acting out whatever your brain is doing?
Because the brain initiates a profound state of active paralysis.
Active paralysis.
Yes. As you enter REM, descending neural pathways from the brainstem send powerful inhibitory signals down to your spinal motor neurons. Your skeletal muscle tone drops to near absolute zero.
So you're totally limp.
Completely flaccid. This paralysis affects nearly all the voluntary muscles in your body. It spares only a very few absolutely critical systems.
Like what?
Your extraocular muscles, which allows your eyes to dart around, hence the rapid eye movements, your respiratory muscles like the diaphragm, so you can continue to breathe, and your sphincters. Everything else is completely offline and immobilized.
So it's like sitting in a high-performance sports car, and you push the gas pedal all the way to the floor. The engine is revving at maximum RPMs, that's your brain and highly active, burning energy, but you have the transmission completely disengaged in neutral.
The tires are disconnected.
The car doesn't move an inch.
The sports car in neutral is a perfect mechanical analogy, but the changes in REM go far beyond just skeletal muscle paralysis. During REM, your body's autonomic nervous system regulation essentially goes into a state of chaos.
Chaos? How do you mean?
Well, during non-REM sleep, your body temperature drops, but your brain still tightly regulates it, much like a thermostat. If the room gets cold, you shiver. During REM, your body essentially abandons temperature regulation.
You stop trying to regulate.
Yes. You lose the ability to shiver if you are cold or sweat effectively if you are hot. You become essentially poikilothermic, like a reptile, reliant entirely on the ambient temperature of the room.
Wow, so you're basically cold-blooded for a few minutes.
Essentially, yes. Furthermore, your heart rate and blood pressure, which were very slow and stable during N3, begin to fluctuate wildly and unpredictably.
So the heart is just racing and slowing down randomly.
Exactly. And, interestingly, men will experience penile erections, and women will experience physiological signs of sexual arousal driven by this autonomic storm, entirely regardless of the actual psychological content of the dream.
It's just a state of absolute physiological chaos.
Yeah.
You have an active brain trapped entirely within a paralyzed, unregulating body.
That sums it up perfectly.
And over the course of an 8-hour night, you don't just stay in one stage. You bounce back and forth between these states, right? You drop down into N3, come back up for some REM, go back down.
Yeah.
But the proportions of those cycles change as morning approaches.
They do. The architecture evolves across the night. Each full cycle from light sleep to deep sleep to REM and back takes about 90 to 110 minutes.
Okay.
Early in the night, during your first few cycles, you plunge rapidly into that deep, slow wave N3 stage, and you stay there for a long time. Your first REM periods might only last 5 or 10 minutes.
So the front half of the night is mostly deep sleep.
Exactly. But as the night progresses, the body's homeostatic need for deep N3 sleep is largely satisfied. So with each subsequent 90-minute cycle, you spend less and less time in N3.
It gets shallower.
Yes. The non-REM stages become lighter, mostly stage N2, and the periods of REM sleep become progressively longer and more intense. The longest REM periods occur in the last third of the night.
Ah, which is why by the time your alarm goes off in the morning, you are very often waking up spontaneously right out of a long, vivid REM episode.
Precisely why you remember those morning dreams so clearly.
Okay, we've got the map of the territory. We know what it looks like electrically and chemically. We understand the stages. But to understand how the brain initiates this massive, coordinated shutdown every single night, we first have to understand the default state that it is interrupting.
We have to understand wakefulness.
Exactly. We have to understand the engine of wakefulness. And to do that, we have to look back at a medical tragedy from a century ago that gave neuroscientists their very first real map of this system.
You are referring to the groundbreaking, though tragic, work of Baron Constantin von Economo.
Yes, the encephalitis lethargica epidemic. We need to set the historical stage for this because it is such a wild story of scientific discovery born out of sheer crisis. What exactly happened right around the end of World War I?
Right around the end of the First World War, spanning roughly from 1916 to the late 1920s, there was a global outbreak of a very unusual, devastating viral infection that specifically attacked the central nervous system.
And this was called encephalitis lethargica.
Correct. Constantin von Economo was a brilliant Viennese neurologist who carefully observed and treated these patients. The presentation of the disease was deeply unsettling.
What were the symptoms?
The primary symptom in the vast majority of patients was profound, unrelenting sleepiness. They were trapped in a state of extreme hyposomnolence. They would sleep for 20 or more hours a day.
But they weren't in a coma, right?
No, and that is the crucial distinction. When they were forcefully roused by the nurses, they were cogent, they were lucid, they understood where they were, but they simply lacked any drive to remain awake.
Wow.
They would only stay awake long enough to chew a few bites of food before falling right back into a deep sleep mid-sentence. It was an overpowering pathological drive to sleep that lasted for weeks or months.
And tragically, the mortality rate for this disease was incredibly high. But von Economo was a meticulous scientist, and he examined the brains of these patients post-mortem. Without modern tools like fMRI or PET scans, what was he able to deduce just by looking at the tissue?
He was looking for the physical footprint of the virus, and he found focal damage-specific localized lesions caused by the inflammation in the brain.
Where were they?
In the patients who had exhibited that profound 20-hour-a-day sleepiness, the damage was consistently located in a very specific area, at the junction of the midbrain and the diencephalon, deep in the upper brainstem.
Okay, the upper brainstem.
Yes. This observation led von Economo to a brilliant deductive hypothesis. He proposed that this specific upper brainstem and posterior hypothalamus area must normally contain the critical neural circuitry that actively stimulates the forebrain to produce the state we call wakefulness.
So if a virus destroys that specific area, the brain loses its internal alarm clock. It simply cannot wake itself up.
Exactly. The engine of wakefulness is destroyed.
But the encephalitis lethargica epidemic presented a second, completely different set of symptoms in a smaller group of patients, didn't it?
It did. A smaller subset of patients in the exact same epidemic suffered from the exact opposite problem. They had severe, unrelenting, agonizing insomnia.
Just total inability to sleep.
Yes, they felt physically and mentally exhausted, but they simply could not fall asleep. They would fall into a fitful, restless doze for maybe a few hours a day, but wake up completely unrefreshed. They were locked in a state of continuous wakefulness.
And what did their brains look like?
When von Economo performed autopsies on the brains of these patients, he found that the lesions were in a completely different anatomical location. The damage was concentrated in the anterior hypothalamus near the basal forebrain.
So the logic is inescapable. Damage to area A in the brainstem causes endless sleep, which means area A promotes wakefulness. Damage to area B in the anterior hypothalamus causes endless wakefulness, which means area B promotes sleep.
That was precisely his logic. He deduced that the anterior hypothalamus must contain sleep-promoting neurons that actively work to inhibit the brainstem's arousal system.
It is amazing he figured that out 100 years ago.
It is truly remarkable about the history of neuroscience. Modern researchers using molecular tracing tools, optogenetics, and functional imaging that von Economo couldn't have possibly imagined have confirmed that his basic architectural model of a wake-promoting brainstem and a sleep-promoting anterior hypothalamus was incredibly stunningly accurate.
Okay, let's fast forward 100 years to today. We know von Economo was right about the general neighborhood. But if we are mapping out this ascending arousal system right now with modern science, how does the brain actually wake itself up? What are the actual neural wires and the specific neurotransmitters do the heavy lifting to keep us conscious?
The modern map of the ascending arousal system reveals a system built on extreme redundancy.
Redundancy, so lots of backups.
Exactly. From an evolutionary standpoint, your brain does not want a single point of failure when it comes to waking up and running from a predator. If one wire gets cut, you still need to be able to wake up. We can broadly divide this massive arousal network into two distinct categories: the modulatory pathways and what we might refer to as the primary power switches.
Let's start with the modulators, the fine-tuning knobs.
The modulatory system relies on a collection of distinct cell groups in the brainstem and hypothalamus that produce different monoamine neurotransmitters. These are chemicals you've likely heard of.
Like what?
First, you have the locus coeruleus, which is a small nucleus in the pons that pumps out norepinephrine. Then you have the dorsal and median raffinuclei, which are spread along the brainstem and produce serotonin.
Okay, norepinephrine and serotonin.
And crucially, you have the tuberomammillary nucleus in the posterior hypothalamus, which produces histamine.
Histamine. Wait, this connects a huge dot for me. This completely explains why taking a first-generation antihistamine for your seasonal allergies, like Benadryl, makes you so incredibly drowsy. The drug is crossing the blood-brain barrier and chemically blocking one of these key wake-promoting pathways.
That is exactly the mechanism of action. Histamine in the brain promotes wakefulness, block the histamine receptors, and you induce drowsiness.
Wow. Okay, so these are the modulators.
Yes. All of these monoanergic neurons, the norepinephrine, serotonin, and histamine producers, send long axons projecting straight up through the hypothalamus and distributing widely throughout the entire cerebral cortex. Their net effect is to increase cortical arousal and alertness.
So they are just bathing the brain in wake-up juice.
Yes. The firing rates of these neurons map perfectly onto our behavioral states. They fire fastest when you are awake and alert. They slow down significantly during the quiet stages of non-REM sleep, and they essentially fall completely silent during REM sleep.
But I want to make sure I'm understanding the hierarchy here. You called these modulators. They are just the fine-tuning knobs, right? They aren't the main power cord providing electricity to the house.
That is a vital distinction, and it was a source of confusion in the field for a long time. Because these monoamine centers correlate so perfectly with wakefulness, researchers initially thought they were the primary drivers of consciousness.
It makes sense to assume that.
It does.
Right.
But experimental science proved otherwise. If you carefully lesion or destroy these specific monoanergic cell groups in animal models, wiping out their norepinephrine or histamine centers, it doesn't permanently stop the animal from waking up.
Really? They still wake up.
They do. They might have a harder time maintaining wakefulness under adverse conditions, or their sleep might be a bit fragmented. But their baseline total daily sleep and wake times remain mostly unchanged. The animal still wakes up and functions.
So it's not the main switch.
No. The one minor exception is that destroying the dopaminergic neurons near the dorsal raphe does seem to cause about a 20% drop in total daily wakefulness. But again, it does not induce a coma. The core engine is still running.
So if destroying the norepinephrine, serotonin, and histamine centers doesn't put you into a coma, what actually does? Where are the true power switches that turn on consciousness?
The true power switches, the anatomical locations where physical lesions actually cause irreversible coma, are primarily governed by glutamatergic, cholinergic, and GABAergic neurons.
Glutamate.
Yes. Glutamate, as a reminder, is the central nervous system's primary excitatory neurotransmitter. Specifically, there are highly concentrated vital glutamatergic neurons located in the dorsolateral rostral pons, in an area that includes the parabrachial nucleus. There are also critical glutamatergic neurons located a bit higher up, in the supramammillary region of the hypothalamus.
So these are the massive relay stations blasting excitatory glutamate up into the higher brain.
Yes. And alongside them, you have large essential populations of cholinergic, meaning acetylcholine-producing GABAergic and glutamatergic neurons located in the basal forebrain.
The basal forebrain.
Right. These basal forebrain neurons receive signals from the brainstem and project them outward to activate the entire cortex. If a patient suffers massive bilateral lesions damage to both sides of the brain in the dorsolateral pons or the basal forebrain, you essentially sever the main power cable.
The lights go out.
Completely. The cortex completely loses its activating signals, and the result is a profound comatose state from which the subject simply cannot be awakened.
It just shuts down.
We also have to mention the lateral hypothalamus, which contains unique GABAergic neurons. While GABA is usually inhibitory, these specific neurons promote wakefulness by actively projecting to and suppressing the sleep-promoting centers. It's a highly complex, redundant, interwoven web of activation.
Which brings up a really important practical clinical application. Let's say you are a neurologist working a shift in the emergency room. A patient is brought in by paramedics and they are completely unresponsive. You have to figure out what's wrong. How do you distinguish between someone who is just in a very, very deep physiological sleep, someone who is in a stupor from, say, drug intoxication, and someone who is in a true coma caused by actual physical structural damage to this specific arousal system we just mapped out?
It is a critical diagnostic pathway that every neurologist relies on. The fundamental principle is that consciousness requires two things: an active ascending arousal system in the brainstem and functioning cerebral hemispheres to receive those signals.
Okay, so how do you test it?
First, you attempt to wake them. If a patient cannot be awakened even by vigorous, painful stimulation like external rub, they are by definition in a coma.
Right.
If they can be partially awakened but fall back into unresponsiveness the moment you stop stimulating them, they are considered obtunded or stuporous.
So they fail the wake-up test.
Yeah.
What's the next diagnostic check to figure out why they are in a coma?
You have to determine if there is a structural injury to the brainstem where those core arousal pathways live. But you can't see the arousal pathways easily without complex imaging.
Right.
However, because those vital arousal pathways sit physically right next to the cranial nerves that control vital basic brainstem reflexes, you test the reflexes. They act as a proxy.
What kind of reflexes?
You shine a light in the eyes to check the pupillary response. You check for specific reflexive eye movements, often by turning the head or using caloric testing. You closely monitor their spontaneous respiration patterns.
What does that tell you?
If you run those tests and all of those basic brainstem reflexes are perfectly intact, but the patient is still deeply comatose, it tells you something crucial. It is highly likely that the physical structure of the brainstem and the arousal system is fine.
Because if a stroke or a tumor had crushed the arousal system, it would have crushed the reflex nerves sitting right next to them.
Precisely. So if the reflexes work, the coma is almost certainly being caused by a widespread metabolic or toxic issue that is depressing the entire brain simultaneously.
Like drugs.
Yes, a massive drug overdose affecting the cerebral hemispheres, or severe hypoglycemia, or a lack of oxygen to the brain. You would immediately follow up with blood tests, toxicology screens, and a CT scan to rule out something like massive bilateral bleeding. The reflexes give you the anatomical map of the failure.
So the brainstem reflexes act as a proxy indicator for the health of the arousal system. That is a brilliant diagnostic shortcut. Okay, so we have mapped out this massive, highly redundant engine of glutamatergic, cholinergic, and monoaminergic neurons constantly blasting the cortex with wake-up signals.
It's a very powerful engine.
Which raises the obvious mechanical question. How does the brain ever manage to turn this roaring engine off? Like, how do we actually fall asleep?
To turn it off, the brain utilizes a highly specialized, localized kill switch. And this takes us directly back to the concept of state transitions and von Economos patients.
The insomnia patients.
Yes, remember the insomnia patients who had lesions in the anterior hypothalamus? Modern neuroscience has pinpointed the exact nuclei in that region responsible for initiating and promoting sleep.
Let's name the biological tranquilizer darts here. What are the key structures?
The primary actors are a cluster of cells called the ventrolateral preoptic nucleus, or VLPO.
VLPO.
Yes, and a nearby group called the median preoptic nucleus, or MNPO. These small structures are densely packed with neurons that utilize the inhibitory neurotransmitters GABA and galanin.
Inhibitory. So their whole biological job is to shout, "Quiet down," to whatever they touch.
Exactly. These sleep-promoting neurons send long projection fibers out to literally every single major node of the ascending arousal system we just spent time detailing.
They wire directly into the wake system.
They do. The parabrachial nucleus, the locus coeruleus, the brachionuclei, the basal forebrain, all of those wake-promoting power switches and modulators receive dense inhibitory connections from the VLPO and MNPO.
So they just shut them off?
Yes. The firing pattern is clear. These sleep-promoting neurons fire very slowly when you are awake. They start firing faster and faster as you get drowsy and sleep pressure builds, and they fire at their absolute maximum rate during deep N3 sleep, especially if you have been previously sleep-deprived.
Wow.
Furthermore, there is a secondary important group of aberrachic neurons located down in the brainstem called the parafacial zone, or PFZ, whose specific job is to target and heavily inhibit the glutamatergic parabrachial nucleus.
Now, I want to stop you here because I have a hard time buying the underlying logic of this wiring. You're describing a system of mutual inhibition, right?
Correct.
The wake system actively inhibits the sleep system during the day, and the sleep system actively inhibits the wake system at night. They fight each other. But if they're just constantly fighting, why wouldn't they just reach an equilibrium?
That is a very perceptive question.
Why wouldn't they cancel each other out and leave me in a permanent intermediate zombie state? Biology is usually messy and gradual. Why would evolution wire the brain like a binary light switch, strictly on or strictly off, instead of a dimmer dial that just slowly fades from wake to sleep over several hours?
It is a profound question. The answer lies in survival. A dimmer dial would be incredibly dangerous from an evolutionary perspective.
Why?
Think about an animal in the wild, or our early hominid ancestors. To survive, you need to be fully, sharply alert to hunt for food, navigate terrain, and evade predators. Alternatively, you need to be fully, deeply asleep in a safe, hidden location to reap the vital metabolic and restorative benefits of sleep. What you absolutely do not want is to spend 3 or 4 hours of your day stumbling around in an intermediate, groggy, half-awake, half-asleep state. That state is biologically useless. You aren't alert enough to defend yourself, and you aren't asleep enough to rest.
That is the exact moment you get eaten by a lion.
Exactly. Being drowsy is the ultimate state of vulnerability in the wild. So the brain evolved to wire this circuit as a flip-flop switch. It is an electrical engineering concept.
Flip-flop switch.
Yes. The mutual inhibition guarantees that the system is inherently unstable in the middle state. It cannot rest in equilibrium. When you are fully awake, the massive arousal system is heavily inhibiting the VLPO. The sleep switch is pinned down.
So wake is winning the tug of war.
Yes. But as the day goes on, biological sleep pressure builds and the VLPO gets slightly stronger. The moment the VLPO gains just a tiny, slight edge in the biological tug of war, it begins to inhibit the arousal system.
And then what?
As the arousal system weakens, its downward inhibition on the VLPO weakens. This release allows the VLPO to fire even stronger, which further crushes the arousal system, which further releases the VLPO.
Oh, it's a runaway cascade effect, a positive feedback loop of inhibition.
Precisely. The system physically and rapidly collapses from one defined state into the other. This elegant flip-flop design ensures that the actual transition from being fully awake to fully asleep takes only a few seconds to a few minutes.
So it's basically instantaneous.
Yes. You spend almost your entirety of your daily cycle definitively locked into one state or the other, minimizing that highly vulnerable transitional downtime.
A biological flip-flop switch. That really is elegant engineering. And from what I understand in the source material, this exact same binary switch logic also applies down in the basement of the brainstem, governing how we switch between the deep, quiet non-REM sleep and the chaotic, paralyzed dreaming state of REM sleep.
It does. There's a secondary, deeply embedded subswitch located in the brainstem that governs the REM transition.
How does that one work?
To drive the brain into REM sleep, you have a specific network of glutamatergic neurons located in an area called the subceruleus region. These work in concert with cholinergic and glutamatergic neurons in the parabrachial and pedunculopontine nuclei.
So these are the REM-on neurons.
Correct. When these REM-on neurons fire, they drive the fast, awake-like EEG rhythms in the cortex. And crucially, they send powerful signals down through the ventromedial medulla directly into the spinal cord to deeply hyperpolarize the motor neurons.
That is the physical mechanism of the REM paralysis switch.
Yes, that is how the transmission gets put into neutral.
But they can't just fire all the time or we'd be paralyzed constantly.
Yeah.
What keeps the REM switch locked down when we don't want it?
These REM-promoting neurons are actively, heavily suppressed by the wake-promoting monoamine systems we talked about earlier, specifically the norepinephrine from the locus coeruleus and the serotonin from the dorsal raphe. When you are awake, these monoamine centers are blasting their chemicals throughout the brain, which completely locks down the REM-promoting neurons in the subceruleus. This intense suppression is exactly why you don't spontaneously drop into paralyzed REM sleep while you are walking down the street or driving a car.
Wait. This perfectly explains a really common side effect of modern pharmacology.
Yeah.
Millions of people take SSRI antidepressants, selective serotonin reuptake inhibitors. These drugs literally work by artificially boosting the amount of active serotonin floating around in the brain. So if serotonin is the chemical that actively suppresses the REM switch,
then taking an SSRI will heavily suppress your REM sleep.
Wow.
Yes. People who begin taking these medications very often experience significantly reduced REM time on a polysomnogram. And conversely, when a patient abruptly stops taking an SSRI, removing that heavy serotonergic blanket of suppression, they frequently experience a massive REM rebound.
What happens then?
The REM switch fires uncontrollably, resulting in intense, overly vivid, sometimes disturbing dreams. The clinical pharmacology maps perfectly and directly onto the underlying neuroanatomy.
I love when the biochemical puzzle pieces just snap together like that.
Yeah.
Okay, so we've mapped the complex architecture of the sleep stages, we've identified the massive, redundant engine of wakefulness, and we've detailed the elegant flip-flop switches that rapidly transition us between the two states.
We have the circuitry built.
But we are missing a huge piece of the puzzle. We haven't answered the most fundamental mechanical question. What is actually pressing the button to flip the switch?
We need to define the biological pressure that drives the transition.
Right. According to the foundational model, sleep is regulated by a two-process model. You have process S, which is the homeostatic drive, and process C, which is the circadian drive. Let's tackle process S, the homeostatic drive, first. How does a brain objectively count how long you have been awake? If there is a pressure building up, what is the actual chemical currency of sleepiness?
The physical currency of sleep pressure is a small molecule called adenosine.
Adenosine.
Yes. To understand how this works, we have to zoom in and look at basic cellular metabolism. When your brain is awake and you are thinking, moving, and interacting with the world, your cortical neurons are highly active. And biological activity requires burning energy.
Right.
The primary universal energy molecule in your cells is ATP, adenosine triphosphate.
ATP, the little molecular battery packs of the cell.
Exactly. As your neurons fire action potentials, they consume ATP. They strip away the three phosphate groups to release the stored energy for cellular work. What is left behind after the energy is spent is the core base molecule, which is dephosphorylated adenosine.
Okay, so it's the leftover piece of the battery.
Yes. As you remain awake hour after hour, continuously burning ATP, this leftover adenosine begins to slowly build up in the extracellular space surrounding the neurons, particularly in the basal forebrain and the cortex. The longer you remain awake, the higher the concentration of adenosine becomes.
So if the brain is an engine, adenosine is essentially the exhaust fumes. The longer the engine runs, the more exhaust builds up in the garage.
That's a perfect visual metaphor. And this adenosine doesn't just sit there inertly. It acts as a powerful local neuromodulator. It physically binds to specific protein receptors located on the surface of surrounding neurons, and it works via two distinct pathways.
Break those pathways down for me.
First, adenosine binds to adenosine type 1, or A1 receptors. These are inhibitory receptors, and they are located extensively throughout the wake-promoting systems we previously discussed, like the basal forebrain and the brainstem arousal nuclei.
So as the exhaust fumes build up, the adenosine physically binds to the arousal system and directly suppresses its ability to fire.
Yes.
It's physically dragging the wake-promoting neurons down, making it harder for them to stay active.
Exactly, but it gets more complex. Simultaneously, adenosine also binds to adenosine type 2A, or A2A receptors.
Okay, A2A.
These are excitatory receptors. They are particularly concentrated in an area of the brain called the shell of the nucleus accumbens, as well as the olfactory tubercle. When adenosine activates these specific A2A receptors, it sends an excitatory signal up to the hypothalamus, specifically stimulating the GABAergic neurons in the VLPO.
The sleep-promoting center.
The sleep-promoting center.
So the exhaust fumes are doing two distinct things at the exact same time. They are taking the foot off the gas pedal by directly inhibiting the wake centers, and they are pressing down hard on the brake pedal by actively exciting the sleep centers.
Precisely. And this dual coordinated action is what creates that overwhelming, inescapable sensation of pressure to sleep after you've been awake for 24 or 36 hours. The flip-flop switch gets pushed relentlessly and unstoppably toward the sleep state.
Which brings us to the most popular psychoactive drug on the entire planet. I pointed this out in our outline prep, and I just find the chemistry so deeply satisfying. Caffeine.
The world's favorite antagonist.
Yes. The caffeine molecule that is in your morning coffee or tea is structurally so similar in its shape to adenosine that it physically fits right into those exact A1 and A2A receptors like a key in a lock. But because it's not actually adenosine, it doesn't turn the lock. It doesn't activate them. It just parks there, blocking the actual adenosine exhaust fumes from binding.
So when you drink coffee, your brain is functionally blinded to its own exhaust.
Yes, from a pharmacological perspective, caffeine is a competitive adenosine receptor antagonist. And the critical thing to understand is that while the caffeine is blocking the receptors, the underlying homeostatic sleep pressure is still physically building up.
And the engine is still running.
You are still awake, you are still burning ATP, and you are still generating massive amounts of adenosine in the extracellular space. But the brain simply cannot feel it because the receptors are plugged.
Right.
However, caffeine has a half-life. Once the liver metabolizes the caffeine and it clears out of the receptors, all of that massively accumulated adenosine rushes the newly freed receptors all at once.
And you experience the classic, sudden, devastating caffeine crash.
You can't cheat the biological ledger. The debt always comes due. Now, adenosine is the primary driver of process S. But are there other chemicals floating around in the body that contribute to this homeostatic pressure to sleep?
There are indeed. We refer to them collectively as humoral factors, and many of them are intimately tied to the body's immune system.
Oh, interesting. Like what?
For example, specific cytokines like interleukin-1 beta and tumor necrosis factor alpha, as well as lipid molecules like prostaglandin D2, all accumulate in the brain and have been shown to actively promote non-REM sleep.
Wait, cytokines, that's what your immune system pumps out in massive quantities when you're fighting an illness.
Exactly. This is the precise physiological mechanism that explains why you feel so profoundly, overwhelmingly sleepy when you come down with a severe viral or bacterial infection, like the flu.
You just want to sleep for days.
Yes, your immune system releases these factors into the bloodstream to mount an attack on the pathogen, but those exact same inflammatory factors crosstalk with the sleep-promoting circuitry in your brain, specifically the preoptic area of the hypothalamus. They force the flip-flop switch into the sleep state.
Why would it do that?
Evolution designed this so that your body is forced to lie still and dedicate its massive energy resources to fighting the infection and healing, rather than foraging or exploring.
It's all connected. The immune system literally hijacks the sleep switch.
Yeah.
So if we are in a laboratory setting, how do researchers objectively measure this homeostatic pressure? I mean, you can't exactly ask a sleep-deprived subject, how sleepy are you on a scale of 1 to 10, and expect to get pristine, reliable data.
You absolutely cannot. Self-reporting for sleepiness is notoriously dangerously inaccurate. Sleep-deprived individuals consistently and severely underestimate how impaired they actually are.
Really? They just think they are fine.
They will tell you they feel fine while they are objectively failing basic cognitive tasks. So, sleep researchers use objective behavioral metrics, the most rigorous and common being the psychomotor vigilance task, or PVT.
How does that test actually work?
It is essentially a test of sustained attention and reaction time, but it is designed to be excruciatingly simple. The subject sits in a quiet room in front of a blank screen and watches for a small visual stimulus, like a light or a counter, to appear.
Just staring at a screen?
Yes. The stimulus turns on at random intervals, usually between 2 and 10 seconds, over a sustained 10-minute period. The subject simply has to press a button as fast as they possibly can the moment they see the stimulus.
That sounds incredibly monotonous.
It is intentionally monotonous. A fully rested brain with low adenosine levels can handle it easily, maintaining a fast, consistent reaction time, usually well under 250 milliseconds.
Okay, but what if they are sleepy?
A brain operating under high homeostatic sleep pressure, a brain saturated with adenosine and cytokines, simply cannot maintain that level of basic vigilance. As sleep pressure mounts, the subject will begin to experience what we call microlapses.
Microlapses.
They will be intermittently very slow to respond, taking over half a second. Or, more terrifyingly, they will completely fail to register the visual stimulus at all for several seconds.
So they just stare right at it and don't press the button.
The brain literally goes offline. The sleep centers briefly overpower the wake centers for a fraction of a second, entirely against the conscious will and effort of the subject.
The sleep pressure literally overrides their conscious intent to push the button. That is terrifying if you extrapolate that to someone driving a car at 70 miles an hour. A 2-second microlapse is a fatal crash.
It happens every day on the highways.
Okay, so process S, the homeostatic drive fueled by adenosine, builds up steadily every single waking hour. But here's the narrative tension of our biology. If adenosine buildup alone dictated when we sleep, we would all be hopelessly asleep by mid-afternoon every single day. The pressure would just get too high by 3:00 p.m. But we don't. We manage to stay awake, alert, and active until nightfall. And that brings us to process C, the circadian drive, the master clock.
Yes. The circadian drive is an independent, internally generated, roughly 24-hour oscillating system. Its primary behavioral function in humans is to actively fight against the accumulating homeostatic sleep pressure during the late afternoon and early evening, providing an opposing alerting signal that keeps us awake.
It fights the pressure.
Exactly. And then, at the optimal biological time late in the evening, the circadian alerting signal rapidly collapses, allowing the accumulated homeostatic pressure to finally take over unhindered and initiate sleep.
The interaction between those two processes is beautiful, and the physical location of this master clock in the brain is just mind-blowing to me.
The master circadian pacemaker is located in a structure called the Suprachiasmatic Nucleus, or SCN.
SCN.
This is a tiny, incredibly dense bilateral cluster of about 20,000 GABAergic neurons located in the anterior hypothalamus. And its location is not an accident. It sits directly above the optic chiasm, which is the physical X shape where the optic nerves from your two eyes cross on their way into the brain.
Okay, I want to slow down and do a very deep molecular dive right here. Because it's one thing to casually say there's a biological clock in your brain, and another thing entirely to explain how a single microscopic biological cell can literally keep time. A neuron doesn't have a pendulum, it doesn't have a quartz crystal or gears. How does a puddle of chemicals inside an SCN neuron know that it has been exactly 24 hours?
It achieves timekeeping through a breathtaking masterwork of genetic engineering, specifically a delayed transcription translation autoregulatory feedback loop.
Okay, that is a mouthful. Let's break that down.
I will walk through the exact molecular machinery because it is one of the great discoveries of modern genetics. Let's look inside the nucleus of a single SCN neuron where the DNA resides. The positive initiating limb of this clock involves two specific proteins called BMAL1 and CLOCK.
BMAL1 and CLOCK.
These two proteins are produced in the cell's cytoplasm. They find each other, link together to form what is called a heterodimer, and then they physically travel back inside the nucleus.
So the BMAL1 and CLOCK tag team enters the vault where the DNA is.
Yes. Once inside the nucleus, this BML1 CLOCK mimesis scans the DNA strand and binds to a highly specific genetic sequence called an E-box motif.
An E-box.
This E-box acts like a master promoter switch. When BML1 and CLOCK bind to it, they turn on the transcription of hundreds of different genes which drive the daily rhythms of the cell. But for the core timekeeping mechanism itself, the crucial genes it activates are the period genes, specifically PER1 and PER2, and the cryptochrome genes Cry1 and Cry2.
Okay, so BMA1 and CLOCK hit the DNA switch, and the factory starts manufacturing the blueprints for period and cryptochrome proteins.
Correct. The DNA is transcribed into messenger RNA, which leaves the nucleus. The ribosomes in the cytoplasm read the mRNA and translate it into actual PER and CRY proteins.
Got it.
But here's where the delay happens. It takes time to build these complex proteins. Over the course of several hours during the biological day, the physical concentration of PER and CRY proteins slowly rises in the cytoplasm. They are piling up.
Like inventory in the warehouse.
Exactly. Eventually, they find each other, dimerize, and form a massive complex with an enzyme called casein kinase 1 delta or epsilon.
So we are basically building a highly specific, time-delayed protein bomb on the assembly line.
That is a very apt analogy. Once this new, massive super protein complex consisting of PDR, CRY, and casein kinase 1 is fully assembled, it translocates back into the nucleus.
It goes back into the vault.
Yes, and here is the punchline of the clock. This complex physically targets the original BML1 CLOCK dimer that started the whole process, and it physically binds to it, knocking it off the E-box on the DNA.
Wait, really? It is a factory that builds products that eventually pile up so high they barricade the doors of the factory and physically tackle the manager who turned the machines on. It completely shuts down its own production.
Yes, it is negative feedback at the genetic level. By knocking BA1 and C off the DNA, the PER-Y complex stops the transcription of its own genes. The cell stops making new PER and CRY messenger RNA. The factory goes dark.
So the supply line is cut. What happens next to keep the clock ticking?
Over the next several hours during the biological night, the existing PER and CRY proteins that are in the nucleus slowly begin to degrade and break down.
They just dissolve.
Yes. The casein kinase 1 enzyme, which is attached to them, helps regulate exactly how fast they degrade, acting like a very precise chemical timer. Once the physical levels of PER and CRY drop low enough, they are no longer present in sufficient quantities to inhibit the DNA.
And then what?
BMA1 and CLOCK are freed from the inhibition, they bind to the E-box once again, the factory lights turn back on, and the entire cycle restarts. The physical time it takes for this full, unyielding loop transcription, accumulation, negative inhibition, and degradation is approximately 24 hours.
That is just... It's staggering. It really is a molecular hourglass.
Yeah.
The proteins build up until they reach the top mechanism, flip the hourglass over to stop the flow, and then the cell just has to wait for the proteins to degrade and run out before it flips again. And this exact genetic dance is happening in every single cell.
It is. Almost all the cells in your entire body possess this genetic clock machinery. Your liver cells, your heart muscle, your digestive tract, they all have these cycling PER and CRY proteins.
Wow.
But if you were to extract those cells and put them isolated in a petri dish, they would quickly drift out of sync with each other. One liver cell might run on a 23.5-hour clock, another on a 24.5-hour clock.
So it'd be chaos.
Yes. The biological magic of the suprachiasmatic nucleus is that its 20,000 neurons continuously communicate with each other through neuropeptides, synchronizing their individual molecular clocks into one unified, massively cohesive master pacemaking signal that dictates time for the entire brain and body.
Okay, but I want to catch something you said a second ago. You said the cycle is approximately 24 hours. The genetics aren't perfectly aligned to the rotation of the Earth, are they? Like, without external input, the clock isn't perfectly accurate.
You are correct. If you take a human subject and place them in a deep cave or a completely dim laboratory isolation suite for weeks with absolutely no light cues, no windows, and no clocks, the average intrinsic rhythm of the human SCN will settle at about 24.1 to 24.2 hours.
Which means every single day, your internal biological clock drifts later by about 10 to 15 minutes.
Yes. Over the course of just a few weeks in isolation, you would become completely desynchronized from the actual solar day outside. You would find yourself naturally waking up at sunset and going to sleep at dawn.
That sounds awful.
And this is not just a theoretical bunker experiment. It is the agonizing, lived reality for many completely blind individuals who lack light transmission from their eyes to their brain. Because they cannot synchronize their clock, they suffer from a debilitating condition called non-24-hour sleep-wake rhythm disorder.
Because they keep drifting.
They constantly drift through the solar day.
Yeah.
One week they're sleeping normally, the next week they are experiencing terrible insomnia at night and crushing daytime sleepiness because their internal biology thinks it is 3:80 a.m., while society is at work at 3:00 p.m.
Which highlights just how unbelievably crucial environmental light is for synchronizing, or what biologists call entraining, the clock. The clock needs a daily reset. But how does light actually get from the eye to this tiny cluster of cells deep inside the hypothalamus? Because we aren't talking about vision here, are we?
No, we are not talking about vision. It uses a dedicated hidden neural pathway. We all learn in basic biology about the rods and cones in the retina, the photoreceptors that allow us to see images, detect motion, and perceive colors. But the circadian system uses a completely different, evolutionarily older set of cells. There is a small subpopulation of retinal ganglion cells, about 1 to 2% of them, that are intrinsically photosensitive.
Meaning they react to light all by themselves, without needing any input from the rods or cones.
Exactly. These specific cells contain a unique photopigment called melanopsin. They aren't looking at the shape of a tree or the color of a car. They are essentially functioning as raw biological luminance detectors.
They just know if it's bright or dark.
Yes, they just measure the overall ambient light level in the environment. And they are specifically highly sensitive to short wavelength blue light, the exact wavelength of a clear daytime sky. When the sun comes up, these melanopsin-containing ganglion cells fire action potentials.
And where do the signals go?
These signals travel down a dedicated physical wire called the retino-hypothalamic tract, which bypasses the visual cortex completely and wires directly, physically, into the core of the suprachiasmatic nucleus.
They just shout, "The sun is up!"
Yeah.
Directly at the master clock, bypassing the visual system entirely.
Yes. And that daily burst of neural signaling generated by morning light physically interacts with the genetic transcription cycle we just discussed. It alters the degradation rate of the PER proteins, effectively moving the clock back those 10 to 15 minutes so it aligns perfectly with the 24-hour rotation of the Earth. It zeroes out the drift.
Okay, so light is the unmistakable day signal. What about the night signal? I know everyone and their mother buys it in gummy form at the pharmacy now, but what is the actual natural biological role of melatonin?
Melatonin is the chemical signal of darkness. It is an endocrine hormone synthesized and secreted by the pineal gland, a small structure situated deep in the brain. But the pineal gland doesn't just decide when to release it. It is tightly controlled by the SCN through a fascinatingly convoluted, multi-step neural pathway.
Oh, convoluted.
Very. The SCN projects to the paraventricular nucleus of the hypothalamus, which then sends long axonal signals all the way down the brainstem and into the upper spinal cord to activate the sympathetic nervous system.
Down to the spinal cord.
Yes. The sympathetic nervous system then sends nerve fibers all the way back up into the skull to innervate the pineal gland.
Wow, it really takes the absolute scenic route through the nervous system.
It is a remarkable circuit. When the melanopsin cells in the eye stop firing because the sun has gone down, the SCN recognizes the absence of light. It alters its firing rate, which triggers this multi-step sympathetic relay, ultimately causing the pineal gland to synthesize and release melatonin into the bloodstream and cerebrospinal fluid.
And what does the melatonin actually do when it hits the bloodstream? I mean, people take it thinking it's a sleeping pill. Does it actively sedate you?
Not exactly. It's a common misconception. Melatonin is not a powerful sedative like a barbiturate or a benzodiazepine. It is a biological timing signal.
A timing signal.
Interestingly, neurons within the suprachiasmatic nucleus itself possess high concentrations of melatonin receptors. So the SCN triggers the release of melatonin, and then that same melatonin binds right back to the SCN.
So like a confirmation receipt.
It acts as a feedback loop to reinforce the circadian rhythm, dampening the SCN's alerting signals and telling the brain and body, yes, it is officially biological night, prepare for the transition. This is exactly why exogenous melatonin, those over-the-counter gummies you mentioned, can be highly useful for regularizing sleep onset timing, especially for treating jet lag when you fly across time zones, or for helping those blind individuals with non-24-hour disorder lock in a rhythm. It's a clock-setting hormone, not a heavy tranquilizer.
That makes perfect sense. Now, there's one massive, glaring logical paradox in everything you have just described about the circadian clock, and I really want to dig into segment 6 of our outline here to resolve it. The SCN uses sunlight to synchronize its 24-hour loop. It fires its action potentials during the day when the sun is out and goes quiet at night.
Yes, the SCN is highly active during the daily light period in all mammalian species studied.
But wait, we humans are a diurnal species. We are awake and active during the day and asleep at night. But rats, mice, hamsters, and countless other mammals are strictly nocturnal. They are asleep during the day and awake and hunting at night. If the SCN fires during the daytime for all mammals because it's rigidly tied to the physical presence of sunlight, how does the SCN tell a rat to go to sleep but tell a human to wake up? How does the exact same molecular clock produce entirely opposite behavioral outputs?
That is a brilliant question, and answering it reveals one of the most elegant evolutionary adaptations in neurobiology.
How does it work?
The answer lies in the fact that the master clock, the SCN, does not directly wire into the flip-flop sleep switches we talked about earlier. It uses a complex intermediary relay system. You can think of it as a biological distribution board that can be custom wired by evolution.
Walk me through the wiring of this distribution board.
The SCN, remember, is primarily composed of GABAergic neurons. This means its direct baseline output is inhibitory. It sends the vast majority of its axonal projections to a small adjacent area in the anterior hypothalamus called the ventral subparaventricular zone, or VSPZ.
SCN to the VSPZ, the master clock to the first relay.
Exactly. And here is where the first critical transformation happens. The neurons in the VSPZ fire in exact antiphase to the SCN.
Antiphase, so the opposite.
Yes. Because the highly active SCN is constantly dumping inhibitory GABA onto them during the day, the VSPZ neurons are forced to be quiet during the day. But when the sun goes down, the SCN goes quiet, it stops releasing GABA, and the VSPZ is released from its inhibition. It becomes highly active at night.
Okay, so the VSPZ is essentially an inversion relay. SCN active in day, VSPZ active at night. It flips the signal.
Next up, the VSPZ then projects its axons to a larger structure called the dorsomedial nucleus of the hypothalamus, the DMH. You can think of the DMH as the great distributor.
The DMH?
It is responsible for integrating circadian signals to regulate sleep, feeding behavior, corticosteroid release, and body temperature. And the DMH sends two critical, contrasting, simultaneous projections down to the sleep-wake architecture we discussed earlier.
It splits the signal.
Yes, it splits the signal perfectly. The DMH sends direct inhibitory GABAergic signals down to the sleep-promoting VLPO. At the exact same time, it sends excitatory glutamatergic signals over to the wake-promoting lateral hypothalamus.
Oh, I see it now. So when the DMH is highly active, it is actively suppressing the sleep centers with GABA and actively stimulating the wake centers with glutamate. It is acting as a massive, coordinated stay-awake command to the whole brain.
Yes, and this intermediary relay system, the SCN to the VSPZ to the DMH, is what provides the incredible evolutionary flexibility between species. The molecular clock in the SCN works exactly the same way in a rat as it does in human. It's a rigid, light-entrained genetic timer.
But the output is different.
Exactly. Through millions of years of evolution, different species have altered how their downstream targets interpret the signals from the DMH and VSPZ. By subtly changing the receptor profiles or altering the specific inhibitory versus excitatory balance in these specific hypothalamic relay nodes, a species can flip its entire behavioral cycle.
That's amazing.
It can occupy a distinct temporal niche dominating the night to avoid daytime predators while utilizing the exact same SCN genetic hardware as a diurnal animal.
So the physical clock mechanism is universal across mammals, but the neurological software interpreting the clock is custom-coded for survival. That is absolutely staggering to think about. And this exact relay system is what we torture when we do shift work or fly across the world. The SCN is aligned to the sun, but we force the DMH to try and keep us awake when it wants us to sleep. It's no wonder jet lag feels like a physical illness. Your distribution board is throwing errors.
Precisely. You are putting the homeostatic drive and the circadian drive into direct, agonizing conflict. The interplay between the homeostatic buildup of adenosine, pushing you relentlessly towards sleep, and the circadian clock rhythmically modulating that pressure via these relays represents one of the most elegantly balanced, complex homeostatic systems in all of biology.
Well, listener, I hope you have enjoyed this masterclass as much as I have. We have completely unpacked the dense neurobiology, the electrical stadium waves of the cortex, the brainstem anatomy of arousal, and the intricate chemical and genetic architecture that orchestrates your daily descent into unconsciousness. We've covered everything from the exhaust fumes of adenosine to the molecular barricades of the SCN, but let me be very, very clear. This is only part one.
We have only mapped the healthy, perfectly functioning system. We have established the baseline.
Exactly. In part two of this deep dive, we are going to explore what happens when this incredibly precise, delicate circuitry breaks down. We will be looking at how your sleep architecture inevitably changes as you age. We will dive into the agonizing neurobiology of clinical insomnia, the severe cardiovascular havoc wreaked by sleep apnea, and the truly terrifying reality of REM sleep behavior disorder, where that crucial paralytic switch in the brainstem we talked about fails, and people physically, violently act out their dreams. We will also explore the fascinating role of a neuropeptide called Orexin and how its sudden autoimmune disappearance causes the chaotic sleep-wake instability of narcolepsy and cataplexy.
It will be a completely different, highly clinical lens applied to the exact same neuroanatomy we have meticulously established today.
It's going to be a wild ride. But before we sign off, I want to leave you with one final, lingering thought to chew on. We have spent the last hour talking about the brain as this massive, highly interconnected organ. The cortex talking to the thalamus, the hypothalamus sending signal to the brainstem, the flip-flopped switches coordinating the whole body. We've treated sleep as a top-down, whole-brain phenomenon.
And behaviorally, for a living organism, it generally is. But there is a fascinating experimental observation buried deep in the source material that challenges that rigid, holistic assumption.
Yes. Researchers found that if you take a tiny physical slab of cortical tissue and physically isolate it from the rest of the brain, disconnecting it entirely from the thalamus, severing it from the arousal system, completely removing it from the influence of the SCN master clock, everything, the pyramidal neurons within that isolated slab will spontaneously, all on their own, begin to oscillate between those slow-wave up and down states.
Even totally isolated from the body, devoid of any external neural commands, the cortical tissue defaults into the fundamental electrical rhythm of deep sleep.
Which forces you to wonder, is sleep truly a top-down, whole-brain command driven strictly by the hypothalamus? Or is it, at its absolute biological core, a fundamental, inescapable, emergent property of the individual neurons themselves? When you get into bed tonight, are you going to sleep? Or are billions of your individual cells just collectively deciding they have finally had enough and shutting themselves down? Think about that tonight as you drift off for your next installment of that 25-year excursion into the dark. We will see you for part two.